the general sequence of cell signaling. First, reception. Typically, a signal molecule binds a receptor. Second, transduction. The receptor gets activated by this binding. This often means the receptor will change its shape. It could even involve a whole series of molecules changing their conformation in something called a signal transduction pathway. This can amplify the original signal. Third, response. There's some kind of response that is going to happen. A portion of DNA that is found in the nucleus getting transcribed, for example, that's a type of response. Let's say we have a steroid hormone that travels through the cell's semi-permeable membrane. Remember, that would mean the steroid hormone is our signal molecule, our ligand. Once inside the cell, it binds to a protein receptor within the cell. Now, the protein receptor is active. The protein receptor travels into the nucleus where the cell's DNA is found. This protein receptor binds to DNA and is involved in getting transcription of a certain gene going, which eventually can be... Now, in the example, the receptor was inside the cell, a cytoplasmic receptor. But it doesn't have to be. Receptors are frequently part of the cell's membrane surface. If a receptor is sitting outside of the cell membrane surface, then the ligand doesn't have to come in. In fact, there could be properties of the ligand that may not allow it to pass through the membrane. The ligand could be hydrophilic, which would make it hard to pass through. Let's mention an example of a cell surface receptor type, a ligand-gated ion channel. Ions normally don't go unassisted through the cell membrane. They are charged after all. See our cell transport video. But a ligand-gated ion channel gives them a way through a channel. But it is controlled. In this example, the channel is closed. But here comes a signal molecule, a ligand. It binds the receptor, which is the channel protein in this case. Now, the channel protein responds by opening. The ions, not the ligand, go through. Once ions get through, the concentration of ions increases in the cell. And you may wonder, okay, so now there are ions in the cell. Why does that matter? Well, the increasing ion concentration can trigger a cellular response. After reaching a certain concentration and stimulating a cell response, the ligand can leave its binding site from the receptor, and the channel can close.